Okay. Good evening, Samist. Okay, so I'm Rahul sir, and today we'll be talking about the specificities of the carbon assimilation cycle. Okay, like how do you produce starch, that is known as a dark reaction in your system. Okay, how do you produce starch, then how do you produce sucrose? So all these things will be discussed today. Okay, so the thing is, that is how the things get underway. So those things are known as carbon assimilation cycle, you can say. So photosynthetic organisms, right, they use ATP. Okay, good evening, Pratham. So what we are doing is we are going to start the carbon assimilation cycle. Okay, so photosynthetic organisms, like how do we, or how do the plants they produce starch? How do the plants they produce sucrose? Why do they need to produce sucrose? Okay, that is known as only the dark reaction. Because in many times the students they make the amateur mistake. They say that dark reaction happens in darkness, light reaction happens in light. So we have learned about light reaction. Simultaneously, dark reaction will take place. Just that dark reaction is independent. That too, it is conditional. It is independent of light because it doesn't depend directly on light. But unless you produce any DPH, you cannot go for reductive anabolism, any kind of synthesis pathway, and produce the much needed, okay, starch and sucrose, because at night there is no supply of light. So the plants cannot order food from Zomato and Swiggy and start eating it, right? So at night, what will the plants eat? How will it facilitate, or how will it sustain its metabolism? That is why the plants they produce starch and sucrose, because by breaking those, they can again go for glycolytic process or glycolysis process that I have taught you. Krebs cycle, generation of more ATP, via the electrochemical gradient, chemosmotic theory, proton motive force, and then again they'll be able to recharge or rejuvenate themselves and their metabolic demands and needs. Okay, so photosynthetic organisms, they use ATP and NADPH produced in the light dependent reactions of photosynthesis to synthesize all of the thousands of components that make up the organism. Okay, now this is possible why? Only because of certain enzymatic, you can say, reactions and pathways which are not formed or not found only in chemoheterotrophs. We are dependent on chemicals which are pre-made and we cannot make it. Like humans, we are chemoheterotrophs. Okay, but they have evolved in photoautotrophs. They can make their own food via light energy equal to h, so they are photoautotrophs. So they are not present in chemoheterotrophs like humans. Okay, but are present in photoautotrophs like plants. Okay, now the plants and other autotrophs, they can reduce the atmospheric CO2 to produce various trioses. Trioses means sugars, okay, and use these trioses as precursors, okay, for synthesis of cellulose, starch, lipids, proteins, okay. So all those things can be produced. They can reduce atmospheric carbon dioxide to produce trioses, then can produce the cellulose in the cell wall, starch, sucrose, okay, lipids, all those things can be produced, proteins in the plant. So that is why the plants, they use CO2. Like, have you seen me teaching you carbon dioxide usage in photosynthesis? No, but the reaction says CO2 plus H2O is equal to C6H12O6 plus, you can say, O2. That is the reaction. So that reaction is a very, very rudimentary reaction, you can say, if you want to understand, because it is not the real accumulation of carbon dioxide for the synthesis of ATP. For synthesis of ATP, carbon dioxide is not needed whatsoever. Okay, only excitation is needed. Excitons, I taught you. Fret, I taught you, you can say, how does the fluorescence never occurs, how does the energy transfer occur, pH2, pH1, okay, complete cyclic, non-cyclic, everything else is needed. But CO2 is needed when already you have produced NADPH, then you can use CO2. Carbon assimilation cycle it is. You are assimilating the carbon via assimilating CO2. Those are known as the dark reactions, can be called as, given in your book, can be called as carbon assimilation cycle. Okay, carbon assimilation cycle, because they're able to cause the accumulation of carbon and then assimilation of carbon to produce bigger compounds, okay, that will then give rise to, you can say, other extremely essential compounds that are needed, okay, that are needed by the plants in their metabolic process, because without proteins, without cellulose, without lipids, none of the cells can survive. Either it's a plant cell, animal cell, protozoan cell, fungi cell, any cell cannot survive. That is the most predominant thing that is needed. Now, how do they proceed? Okay, how do they proceed? So, green plants contain in their chloroplasts the enzymatic machinery that catalyzes the conversion of CO2 to simple organic compounds, a process known as CO2 assimilation. Okay, this process is also known as CO2 fixation or carbon fixation. Okay, there are multiple names, or carbon fixation. Okay, so multiple names are present. Okay, now this simple product, the simple product is you can say, just if you have studied glycolysis that I had taught you, now when they reserve the term for the specific reaction of CO2 that is incorporated, okay, into three carbon organic compound, okay, and something, a great triose is produced, okay, known as triose phosphate, meaning that it will contain three carbons only, that is known as 3-phosphoglycerate. Okay, this 3-phosphoglycerate is produced. Okay, and this 3-phosphoglycerate, if you remember, is again produced in glycolysis from 1,3-bisphosphoglycerate. That is the first substrate level phosphorylation event where two ATPs are produced in glycolysis. So here the plants are producing this specific molecule known as the first triose phosphate is being produced, that is known by the name as 3-phosphoglycerate. 3-phosphoglycerate molecule. Okay. Now what happens is this entire reaction of production of, you can say, which culminates into production of 3-phosphoglycerate and later down the line the entire carbon assimilation cycle. This was elucidated in the early 1950s by Melvin Calvin, Andrew Benson, and James A. Basham. Okay, and is also called as the Calvin cycle or photosynthetic carbon reduction cycle. Okay, so Melvin Calvin, Andrew Benson, and James A. Basham, they are accredited for discovering this cycle, also known by the name Calvin cycle. Calvin cycle, okay, or C3 cycle. I tell you why it is called C3 also, and photosynthetic carbon reduction cycle. Carbon reduction cycle. Okay, so this is the overview of why we need to go for even formation of, you can say, sucrose, starch, and all. What is carbon assimilation? Why does the plant need, or why do the plants are in dire need of carbon dioxide? Okay, so that is the main thing behind this process. Okay, so the thing is, the key enzymes that we will see in this process, okay, what they do is, in addition to the universal process of glycolysis, and there is one more process that when you have pyruvate, you can convert the pyruvate back to glucose. That is not in your syllabus, known as gluconeogenesis. Okay, gluconeo, new glucose genesis. So in addition to all this, plants have the unique reaction sequences for reduction of CO2 to triose phosphates. That entire pathway, those enzymes are absent in our system. Okay, we cannot go for CO2 accumulation or CO2 assimilation, I should be saying. Those pathway, those enzymes are absent. Okay, in our body. Otherwise, we could have taken in carbon dioxide and produced starch in our own body. Okay, that is how you can differentiate an animal cell from a plant cell, because those enzymes are, the genes encoding those enzymes are absent in our genome. Okay, so you can think of genetically engineering. Okay, but that will take lots of years. Okay, if you want to put that genome and then see how does it interfere with the other human metabolic needs and demands. Okay, so that can be done, but it will take a lot of time. Okay, for testing, clinical trial, and all. So the thing is, they are very coordinately regulated, okay, to ensure proper allocation of carbon to energy production and synthesis of starch and sucrose. Okay, so what we have is, we have reduction of disulfide bonds, okay, by electrons flowing from photosystem 1, PS1, P700, change in pH and Mg2 plus concentration. Okay, those things will happen. Okay, so covalent modification of, you can say, certain in enzymatic reactions, allosteric regulation. Allosteric regulation means If any enzyme is present, you have an active site and we have one other site in that enzyme as well. Okay, it is very, very important for regulatory pathways. If you have an enzyme like this, enzyme is the lock, substrate is the key, this is the active site. You can have a different site here. If some enzyme binds here, this is known as the allosteric site for the enzyme. If some molecule compound binds here, okay, what it can do is it can have consequence here. It can it can stabilize the active site so that it can bind to substrate better. If this molecule stabilizes the active site and enables it to bind to the substrate in a better manner, that compound will be known as allosteric activator. It activated the enzyme for enzyme induced reaction catalysis. But if the enzyme is an allosteric inhibitor, it will also bind to its allosteric site but will destabilize the active site and substrate won't bind. And no reaction would happen. Okay, so those kinds of things are also present in plants as well as in humans. Okay, so number one thing, okay, that I'll tell you about, okay, the reductive process is the presence of a specific, okay, enzyme known as ribulose 1,5-bisphosphate carboxylase oxygenase. Okay, this enzyme is written in capital letter, bold letter in your NCRT book and all your reference books. Okay, this is an irreplaceable enzyme, such a great enzyme that we'll be talking about it in detail. Okay, the first enzyme that we humans lack, the most important enzymes that plants possess, is ribulose 1,5-bisphosphate carboxylase oxygenase. Short form for this, acronym for this is RuBisCo. Okay, is known by the name as RuBisCo. It's a huge enzyme. Okay, the name is also too long, too lengthy, so thereby scientists have provided it a proper acronym. Okay, in order to remember the name known as RuBisCo. Okay, so what it does is, okay, the carbon assimilation cannot occur without this RuBisCo. Okay, so first stage is there is something known as already a compound known as ribulose 1,5-bisphosphate. Means bisphosphate and biphosphate, you already know the difference. Okay, completely. Bisphosphate means at first position phosphate, fifth position phosphate. So I'm calling it 1,5-bisphosphate. So when you have to assimilate carbon, means you have to take up the carbon. Without this enzyme RuBisCo, the carbon assimilation cannot take place. Means the first reaction of the C3 cycle, okay, the very first reaction of the C3 cycle is what? Is ribulose 1,5-bisphosphate, okay, plus carbon dioxide. This is a 5 carbon, this is a 1 carbon compound. Okay, what you will get is 2 times 3 carbon compound known as 3-phosphoglycerate. It is also producing glycolysis, an intermediate of glycolysis that we have already talked about during the first substrate level phosphorylation event. Now you get this reaction. Okay, this is given in your book also. Okay, so 3-phosphoglycerate. Now this entire reaction can occur due to the cofactor Mg2+, but cofactors can only activate enzymes. They by themselves they will not cause any reaction. So that enzyme necessary is RuBisCo. This is the first and the most important reaction in the C3 cycle. Okay. Calvin cycle. Okay. Now, it is on a C3 cycle? Why? Because the first intermediate produced 3-phosphoglycerate is of how many carbons? 3 carbons. That is why it is also known as C3 cycle. Okay. But that is why it is also known by the name as C3 cycle. Okay, 3-phosphoglycerate. Then that is the formation of a 3-carbon compound known as C3 cycle. Okay, and this reaction is impossible without the presence of, you can say, RuBisCo enzyme. Now let's consider RuBisCo enzyme in detail. Let's talk about it in detail little bit. Why is it important and why is it the most important enzyme in the world? Okay, it is also considered as the most important enzymes, okay, in the world, and all the enzymes which take part in carbon assimilation, very important, are present in the chloroplast stroma. There are total 13 enzymes, okay, which are in this. So all the 13 enzymes that take part in carbon assimilation reactions are present in the stroma, which is equal to the matrix of the mitochondria. Okay, are present in the stroma of chloroplast. Okay. Now let's talk about RuBisCo in detail. Okay, deductive, you can say, photosynthetic carbon assimilation cycle. Now, since the name of the enzyme is ribulose 1,5-bisphosphate, just look at the name first. Okay, by name only you can, by analyzing the name, you can come to two conclusions. When we talk about RuBisCo, ribulose 1,5-bisphosphate carboxylase oxygenase, that is the enzyme. Now, just look at the name. It has been given in the name only carboxylase and oxygenase. Means it has got a dual activity. Sometimes it can cause addition of carbon dioxide to ribulose 1,5-bisphosphate, but once in three times, once in three events, it can cause addition of oxygen also to RuBP, that is ribulose 1,5-bisphosphate, but mostly it will cause addition of carbon dioxide to RuBP. So it is a dual specific enzyme. It can cause the addition of various substrate. One is CO2 with RuBP, that is ribulose 1,5-bisphosphate, or O2 with RuBP. Again, one of the substrate is constant, ribulose 1,5-bisphosphate, but CO2 and O2 can alter in mostly 3:1 ratio. 3 times CO2, 1 time O2, like that. Okay. Now, first things first. Now, what it has been able to do is there are distinct forms of RuBisCo. Okay, form 1 is found in vascular plants, algae, and cyanobacteria. Form 2 is confined to photosynthetic bacteria only, like purple bacteria, green sulfur bacteria. Okay, so neat question is there are two types, okay, of RuBisCo present. The form 1 is present in vascular plants, xylem phloem ones, okay, algae and blue-green algae, cyanobacteria. Form 2 is present in bacteria only. Okay, like we talked about the purple bacteria P870, green sulfur bacteria P840, so they are present there only. Okay. Now next up, what we can talk about with respect to RuBisCo is the plant RuBisCo, the crucial enzyme in the production of biomass CO2. It is a form 1 structure. Okay, it has got 8 identical large subunits. Okay, huge subunits, and each subunit contains a catalytic site, means a kind of active site where reaction can happen. Okay, each subunit can it contains a catalytic site. Okay, and you can say the plant enzyme has an exceptionally low turnover number. Okay, low turnover number means it has very low Vmax. You can say once we have talked about enzyme, we have talked about Km value and Vmax value. So it is able to produce very less amount of substrate. The rate of reaction is very low. It has got a very low turnover number. You can say so. Only 3 molecules of CO2 are fixed per second per molecule of RuBisCo at 25 degrees Celsius. Means standard temperature and pressure. Pressure is 1 atm. So RuBisCo from the comparative level has got a very low turnover. There is no turnover, means very less turnover. Okay, very less number of products you are getting. Okay, a very low number of products you are getting. So very low turnover number. Okay, low Vmax, high Km. Okay, these are the reasons. Affinity is less. Okay, for its own substrate. Therefore, reaction velocity takes time to be reached, and then turnover number will be adversely affected. Means lesser amount or lesser number of product molecules generated per second. Okay, so what I told you was it has got a very low turnover number with the formation of only 3 molecules of CO2 it can take in, and you can say it can fix only 3 CO2 molecules per second. It has to react with 3 RuBP, ribulose 1,5 bisphosphate. Okay, ribulose 1,5 bisphosphate, 3 RuBP. Okay, at 25 degrees Celsius. Okay, at 25 degree Celsius per molecule of RuBisCo enzyme at 25 degree Celsius. That's its capacity. That is its only capacity. Very low turnover number. Okay. So this thing keep in mind always. Also, since this has a very low turnover number, this will create another concept, another consequence, that in order to achieve high rates of carbon dioxide fixation, okay, in order to achieve high rates of carbon dioxide fixation, plants would therefore need more and more amount of this enzyme or not, because one enzyme is only able to produce 3 CO2 molecules, or fix 3 CO2 molecules, means produce 3 phosphoglycerate molecules per second, which is a very low turnover number. Considering any enzymes where 500, 600 is, you can say, turnover is also available. Okay, at least 100 molecules are also available. So 3 molecules per second is a very low turnover number, and the plants are huge. You can say most of the plants that you see angiosperms and all, they are huge. They will need a high amount or a gargantuan amount of starch to be produced, lipids, proteins to be produced, but for that carbon dioxide must be assimilated and fixed. So in order to achieve high rate of CO2 fixation, plants therefore require large amounts of this enzyme. Okay, in order to fix large number of CO2 molecules Plants therefore require, okay, you can say large amount of this enzyme. You can call it high rate also. So large amount of this enzyme is needed, and you know how many plants are there. Plants are so huge creatures. So you can say that RuBisCo makes up almost 50% of the soluble protein, because this is an enzyme and it is a protein too. It is not an RNA, not the ribozyme that we're talking about. This is a protein class enzyme. So RuBisCo makes up almost 50% of the soluble protein in chloroplasts. Okay, 50% of the soluble protein in chloroplasts, and therefore one can say with conviction that it is probably one of the most abundant enzymes in this entire earth or biosphere. This is last thing is more important. This is one of the most abundant enzymes in this biosphere. Now, in NEET examination, there will be a question which is the most abundant enzyme on this or in this biosphere. You will say RuBisCo only. Okay, not one of the as per NEET. This is the most abundant, but the abundance is decreasing because we are going for more and more deforestation. So now the specificities may change a bit, but technically, if plants work in the number that they used to be, this is the most abundant enzyme. Okay, in this biosphere. Okay, most abundant enzymes is the number one enzyme with respect to concentration and you can say the availability in the biosphere. Okay, so next is the specificities about RuBisCo that we have been able to discuss, and it has got a great tertiary structure. Okay, lysine residue is there that will cause the, you can say, carbamylation reaction, but those things are not present. Magnesium is present at the active site, so the entire mechanism is not present. Okay, it is just that there is a carbamylation. Okay, mechanism is the plant RuBisCo is a carbamylated site, lysine side chain it has, it is bound to magnesium ion. Okay, the magnesium ion brings together and orients the reactions at the active site. Okay, causes the polarization of the CO2 and opening it for a nucleophilic attack by the 5 carbon RuBisCo. Okay, so those things are not there, so we'll not go much into the detail about the reaction mechanism, but it has got a great reaction mechanism. Once you, if you become a biochemical or biochemist or a biochemical scientist, then you will be exploring these things later on in life. Okay, now the thing is, you can say that ribulose 1,5-bisphosphate, okay, sometimes can bind very tightly, okay, to the active site and lock the enzyme in the closed conformation. This one information I'll give you, that ribulose 1,5-bisphosphate can sometimes bind too tightly. Okay, too tight reaction is also not good in biology. That if you bind too tightly, you are locked with the, you can say, active site of the substrate. Then the product, when it is formed, this cannot release the product. Okay, the reaction may not occur. Too tight binding and too light binding, both are, you can say, problematic. Now, what happens is, this gives you a closed conformation. The conformation becomes closed. Okay, suppose you have ribulose 1,5-bisphosphate. trying to bind to the RuBisCo active site but too tightly. The thing is, when you are bound with it too tightly, that will make this conformation from open conformation to completely closed conformation. Closed means now it cannot convert this into product. Okay, CO2 induced, you can say CO2 cannot come in, even if it has bound, it is so tightly bound to, you can say RuBP or ribulose 1,5 bisphosphate, it's another substrate, that this cannot mount any kind of nucleophilic attack. Okay, and thus cannot cause any kind of reaction. Okay, that is the problem. Now what happens is, there is another enzyme known as RuBisCo activase, which overcomes this inhibition by promoting ATP dependent release of ribulose 1,5 bisphosphate. Means one ATP is used up here, okay, to produce, or you can say to release the ribulose 1,5 bisphosphate. Very, very important later down the lane, because when you will write the summary of the reaction, this ATP will be used. Now here ATP is catalyzed, ATP is broken down by the enzyme, an enzyme known as RuBisCo activase. ATP is either hydrolyzed to ADP plus Pi plus energy, and this energy will change this closed conformation to open conformation. So energy will be required, from where will you get? From ATP hydrolysis, and then ribulose 1,5 bisphosphate will be released from the tight binding. Okay. Released from the tight binding at the active site. Okay, so this is a great event, that's why I told you, because in the end, in the books that you will study, you will see one more ATP gets added. Okay, now you will wonder from where did it get added. Here that is the answer. Okay, but this answer is not given in your book. They have just written one more ATP gets added in the last step. So this is the real reason behind, okay, the formation. Okay, now let's come to, you can say, the understandings of this C3 cycle. It is just the reverse of glycolysis once we have been able to produce 3-phosphoglycerate. Okay, so quite simple it is. So basically, okay, once we study it in, for, you can say, in the simpler way, then we'll see the, you can say, molecular dynamics. So you have ribulose 1,5 bisphosphate reacting with CO2. Okay, now you have been able to form 3-phosphoglycerate. Okay, 3-phosphoglycerate by the enzyme known as RuBisCo. Okay, by the enzyme known as RuBisCo. Now, if we talk about the reaction, okay, if we talk about the reaction, so ribulose 1,5 bisphosphate, if you talk about, okay, so it looks quite huge. Okay, like not very huge, but it looks quite, the structure is this one, you say CH2. This is the first carbon only, so 1,5 bisphosphate. So I have added up, P means phosphate group. Okay, P means this one here. Okay, PO4 3 minus. Okay, that is the meaning of P. Okay, of phosphate. Wherever I write P, that means I'm writing this phosphate group. Okay, not complicated. I'm just writing P. Okay, so next carbon will be, this is the Fisher projection. Okay, then CHOH. then again CHOH, then I can write again CH2OP, carbon 1, 2, 3, 4, 5. So this is 1,5 bisphosphate plus when you have added CO2, what you will get is 2 times 3 phosphoglycerate. So that is, you can say COO minus, okay, then CHOH, then you can say CH2OP. This is your 3 phosphoglycerate. You can see one phosphate is here, another phosphate will be in another molecule, 2 molecules will form. So number of phosphates here 2, here will be 2 because 2 molecules. At the third position, third carbon, 1, 2, third carbon, 3 phosphoglycerate, okay, is produced. This is the first reaction. And this reaction is catalyzed by RuBisCo. Okay, this reaction is catalyzed by RuBisCo itself. Okay, and where does it happen? In the stroma. Okay, in the stroma of chloroplast. Okay, always remember it happens in the stroma of the chloroplast layer. Now what will happen is we would need one ATP. We are going opposite of glycolysis. Okay, we would need one ATP. Okay, to produce 1,3-bisphosphoglycerate, and this is a reduction process because we are requiring an ATP, and after this the reduction process will come in. So first of all, here substrate level phosphorylation used to happen. So we used to get 2 ATPs. So here we will be needing 2 ATPs to convert 2 phosphoglycerate to 1,3-bisphosphoglycerate. Just the opposite of glycolysis, you can say. We are going in the opposite way because we are going for reduction. Okay. So 3-phosphoglycerate will be converted to ATP is required now. Now this Pi will be attached to form 1,3-bisphosphoglycerate. Okay, 1,3-bisphosphoglycerate is produced. Okay, again the enzyme will be, you can say, same 3-phosphoglycerate kinase. So enzyme is kinase only. Okay. Now 1,3-bisphosphoglycerate is produced. If you had drawn the structure, if you want to see the structure again, I draw it for you. Okay, that is COO. 1,3 means at first position one phosphate I have put. Okay, then CHOH, then CH2OP again. So this is 1, 2, 3 carbon number 1,3-bisphosphoglycerate has been produced. All these reactions are occurring in the, you can say, stroma of the chloroplast. Okay, always remember, in the stroma of the chloroplast. Next will be 1,3-bisphosphoglycerate. Okay, will be reduced to form glyceraldehyde 3-phosphate, 2 glyceraldehyde 3-phosphate molecules. Okay, they will be producing 2. So here we are going 1 molecule each. So I'm just mentioning 1 molecule, but it will be 2, 2, 2 always. Okay, you have to maintain the stoichiometry. So 1,3-bisphosphoglycerate, okay, undergoes reduction. Okay, undergoes reduction. 1,3-bisphospho. Glycerate undergoes reduction by the enzyme dehydrogenase, because dehydrogenases can cause both the reactions, oxidation and reduction, because they fall under the category of oxidoreductases. Okay, when I taught you enzyme, I told you they fall under the category of oxidoreductases. So if the concentration, it depends on the concentration. So if the concentration of 1,3-bisphosphoglycerate is more reactions, then as its concentration is more, so it will go in the reverse direction. Okay, so here, since it is reduction, something will be oxidized. NADPH plus H plus, you will produce NADP plus nicotinamide adenine dinucleotide phosphate. You can say derivative of NAD plus only. Used in plants more, use NAD plus more, and plants use NADP plus more. Okay, NADH, plants use NADPH more. So the thing is that will be converted to, you can say, glyceraldehyde 3-phosphate. Okay. 3-phosphate. But now here 2-pi will be released, just like I told you, because here how many are there? 2 phosphates, here 1 phosphate. So 1-pi will be released as a free pi into the stroma. Because in glycolysis, which was happening in the cytosol, from this radial 3-phosphate, we made 1,3-bisphosphoglycerate. And there I told you 2-pi is taken as a pi is taken from the free reserve of phosphate in the cytoplasm. Yes, you can say pi will be released. If these are two molecules, 2-pi will be released and 2-glyceraldehyde 3-phosphate molecules will be produced. Okay, and now the enzyme, you can say aldolase and isomerase will come in. Okay, first isomerase will come in. We know the structure already. Okay, so first isomerase, it will produce dihydroxy acetone phosphates. These structures are drawn in the glycolytic cycle, so I'm not redrawing it again. Okay, so glyceraldehyde 3-phosphate, and now enzyme aldolase will come in. Okay, just the opposite of glycolysis, to produce what? To produce fructose 1,6-bisphosphate or not? To produce fructose 1,6-bisphosphate. We are just going opposite of glycolysis from 3-phosphoglycerate event. Okay. Now fructose 1,6-bisphosphate, once it has been produced, okay, a new enzyme will come in known as fructose 1,6-bisphosphatase. Okay. So CH2OP, fructose 1,6-bisphosphate. Okay, then CO, and CHOH. Okay, Fischer projection 1, 2, 3, 4, 5, then 6 carbon. Okay, now fructose 1,6-bisphosphate, which is this one. Since we are going the reverse process, the reversal of glycolysis in the reverse orientation, reverse direction, so you need to lose this first phosphate, this one. So what will you do? You will use an enzyme known as phosphatase, which will take out, which will dissociate this bond. Okay, this bond, phosphoanhydride bond, which is a covalent bond, will be dissociated using a phosphatase, fructose 1,6-bisphosphatase. Okay, so simply an enzyme phosphatase will be used. Phosphatase, and that will convert fructose 1,6-bisphosphate to fructose 6-phosphate. Okay, means that CH2OP there will be converted to CH2OH. That's all. This will be converted to, first carbon will be converted to CH2OH. Rest the structure will be completely same. And then after various reactions, okay, various reactions, this fructose 6-phosphate will be converted to starch. Okay, in the next class I'll tell you those reactions. Fructose 6-phosphate will be converted to starch. That is produced in the stroma of chloroplast, always know. stroma of chloroplast. Now, one thing I'll tell you that starch is a storage polysaccharide. The movable polysaccharide is transported throughout is the sucrose. So what happens is the dihydroxyacetone phosphate which was produced, it is exchanged. Okay, the dihydroxyacetone phosphate that has been produced, okay, is exchanged for phosphate. It is exchanged for phosphate. There are the dihydroxyacetone phosphate that is produced is exchanged of phosphate. Why? Because the inner chloroplast membrane is impermeable to most phosphorylated compounds. You can note this down. The inner mitochondrial membrane, the inner chloroplast membrane is impermeable to most of the phosphorylated compounds. Okay. Now, since it is impermeable to most of the phosphorylated compounds, including fructose 6-phosphate, glucose 6-phosphate, and fructose 1,6-bisphosphate, okay, it is impermeable to these. But it does, however, have a specific antiporter that catalyzes one-for-one exchange of Pi with a triose phosphate, not with this 6 carbon phosphates, but with triose phosphate, like either dihydroxyacetone phosphate or 3-phosphoglycerate. Okay, either one Pi will be exchanged with either dihydroxyacetone phosphate or 3-phosphoglycerate. This is known as a triose phosphate. Okay, there is an antiporter, means Pi will be taken in, okay, into the stroma, and dihydroxyacetone phosphate will come out of the stroma. Dihydroxyacetone phosphate. Now, since this is the event that is happening, okay, this antiporter is needed. Why? Because it simultaneously moves Pi into the chloroplast where it is used in photophosphorylation. Otherwise, how will you produce ATP in the Pi? Sorry, in the chloroplast? Because ADP must be phosphorylated with something, with Pi, then you can form ATP. Even if you have produced electrochemical gradient that we have seen in the electron transport chain, you need to react ADP with Pi, otherwise ATP will not form. So that is why Pi is taken in, okay, which moves the triose phosphate into the cytosol and Pi into the stroma, okay. And since it is moved into the cytosol, okay, then this proves one more evolutionary process is this dihydroxyacetone phosphate, okay, will take the same route in the cytosol as well. Okay, it will be converted to glyceraldehyde 3-phosphate by isomerase, half of it. Okay, glyceraldehyde 3-phosphate. This is the cytosol. Okay, this is the stroma of the chloroplast. Now they'll combine both by aldolase, produce fructose 1,6-bisphosphate, Okay, that will be produced eventually. Then again bisphosphatase will cleave it. Again phosphatase, fructose 1,6 bisphosphatase, phosphate ase means cleavage, will convert it into fructose 6 phosphate. And since you have been able to produce fructose 6 phosphate that you produced in the stroma also, but here other some other cytosol, you can say specific enzymes will be present which will convert this fructose 6 phosphate to sucrose, not to starch. And this is transported through the, you can say, phloem that carries food, as they say. Food is sucrose only. Then that is transported through the phloem. Okay. So this is why we go for carbon assimilation C3 cycle. If 3-phosphoglycerate was not assimilated, okay, we would never be able to produce sucrose or starch. Okay, and this pi antiporter, okay, this antiporter plays dual role, as this pi will be used to phosphorylate ADP by F1F0 ATP synthesis present in the in the chloroplast, that will provide this pi. Otherwise, even with electrochemical gradient, you cannot produce ATP because you don't have pi, by what you will be phosphorylating your ADP. Okay, so it forms dual role. It will let the synthesis of sucrose to occur and also formation of ATP. So their evolution has played a great role. Okay, so I hope you were able to comprehend the lecture. If you had any doubts, you can contact me on my personal number. Okay then, thanks a lot.